In the previous unit, we have seen some of the basic mechanisms that can explain genome evolution, like the duplication of exons, the duplication of genes, and the duplication of whole genomes. As I said, unequal recombination events were probably the major molecular process involved in such duplications, but I also mentioned that one of the enabling features of genomes was the presence of large introns and intergenic regions full with transposons and retrotransposons. Now in this unit, I would like to present a couple of very beautiful examples of the action of retrotransposons in shaping genomes. And then I will go on to focus on some recent advances in genome biology that shed new light on potential molecular mechanisms of genome change during evolution. So first, retrotransposon-mediated transduction. I have already referred to this in a previous unit, but it basically means that when a mobile element moves to a new location in the genome, it can carry with it some of the sequence flanking the element. That is known as transduction. It is estimated that up to 1% of the actual sequence of the human genome is the result of transduction events mediated by L1 retrotransposons, a type of mobile element that is very abundant in our genome. And several studies have shown that this mechanism also created gene families. A study published in 2006 showed that transduction mediated by a particular type of retrotransposons called SVA elements was responsible for the appearance of a family of genes in the human genome, and this family includes 4 genes that code for an enzyme called AMAC1. Searching the human genome for sequences that had been transduced by SVA retrotransposons, scientists found 3 transduced sequences located on chromosomes 8, 17, and 18, which apparently originated, all 3 of them, from the same source sequence located on a different place in chromosome 17 as well. The ancestral copy is the AMAC1L3 gene, which had 2 exons separated by an intron. When a SVA element got inserted upstream of this gene and the gene was expressed, the intron was spliced out and the resulting transcript was transposed to 3 other sites in the genome. And that is why the 3 transduced copies of AMAC1L3, which are known as AMAC1, AMAC1L1, and AMAC1L2, lack that intron. They have the remnant of a poly-A tail, and they are also flanked by target site duplications which are formed during the insertion of a retrotransposon. And another great advantage of this type of studies is that you can actually date these events by comparing the genomes of different animals. In this case, scientists found all 4 copies of this gene family in humans and in African great apes, chimps, and gorilla, but not in orangutans, which are non-African great apes. So these transductions must have taken place between 8 and 14 million years ago, after orangutans split from African great apes. There are many other examples of genomic changes mediated by transposons. For example, it is believed that the duplication in the gamma globin gene that led to the gamma 1 and gamma 2 globins in humans resulted from an equal crossing over event in an L1 retrotransposon. But now I just want to introduce another mechanism by which mobile elements contribute to genome plasticity. It is called exonization, that is the formation of a novel exon, and this particular example is very beautiful because it involves a very distant ancestor of humans. The coelacanth is an amazing creature. We knew fossils of coelacanths about 340 million years old, and this fish is believed to be extinct, or it was believed to be extinct until some fishermen back in the 1930s found living coelacanths. The genome of this animal shows why it has changed so little since the time when the ancestors of tetrapods invaded the land. But that's a different story. One very interesting bit of information that scientists also found in the Coelacanth's genome was a retrotransposon that shared about 80% identity at the nucleotide level with two regions of the human genome. One of those regions included an alternative exon in a gene called PCBP2 in chromosome 12. And the other homologous region was an enhancer, a regulatory element in a different gene called ISL1, which is located in chromosome 5. So it is amazing that a small chunk of a mobile element, a retrotransposon from a very ancient type of lobe-finned fish, was later co-opted and used in our genome to perform at least two different functions. On the one hand, it became a new exon in one gene, perhaps creating a new protein isoform with a few more amino acids, whatever, but it also contributed to fine-tune the expression of another gene, becoming an enhancer element. So if you consider the thousands of copies of mobile elements that have been jumping around genomes for millions of years, you will understand how important these elements might have been in genome evolution. In fact, we're just beginning to realize that the scale of this extraordinary process is probably much greater than we had imagined.